Let's just give people a minute to join. I see a lot of attendees still coming in. All right. I think we can start. Hello, everyone. Good morning, good afternoon, good evening, wherever you're joining from. My name is Vishal. I'm a principal architect for R&D at Quantify, and it's my pleasure to welcome you all to day 2 of Conference 2024, our virtual symposium where we're going to cover some of the interesting research from our R&D work. Today on day 2, we are going to predominantly focus on different aspects of our healthcare and life sciences research. So if you've already joined us in the previous session, I hope you're enjoying the event so far. Thank you for your continued participation. But if this is your first session, a warm welcome again from all of us. We're excited to bring you some interesting stuff your way. In the second session for today, we're going to talk a little bit about molecular dynamics. So Temthon in the previous session has just talked about how AI is transforming every stage of drug discovery process. This session will talk about the additional transformative impact of protein engineering in drug discovery. So as we explore how recent advancements like AlphaFold and ESM-fold have revolutionized protein structure prediction, we'll discuss some synergy of these tools with molecular dynamic simulations to enhance our fundamental understanding of biological states, helping in streamlining the design process and ultimately improving the efficacy of therapeutics overall. So to talk more about this, I have my extremely talented and good friend, fellow principal architect by day and a stand-up comedian and an amazing dancer by night, Vishal Pagidipally. So before I hand it over, please note that if there are any questions during the session, please feel free to ask them via the Q&A section at the bottom of your screen. We'd be happy to take them at the end. Over to you, Pali.
Cool. Thank you. Let me just share my screen. Yeah, I hope you can all see the screen that's coming up now. Yeah. So my name is Vishal Pagidipally, and I'm a principal architect in our R&D department. So I predominantly focus on our life sciences capabilities. So what I'm going to talk about is how we use all of the tools that we have out there, like AlphaFold and molecular dynamics simulations, to unlock a new aspect of protein engineering. So I'll just start with a high-level overview of like what protein folding problem is, and then like talk about the AlphaFold and the MD pipeline that we have. One thing that we know, or most people know for now, is most of the pharmaceutical drugs that are out there predominantly act by targeting proteins, and they regulate the protein activity by either upregulating, downregulating, or any kind of modification that they do to the protein activity itself. And we know that the proteins have this ability to fold into integrate 3D structures, and depending on what kind of function and then where they are present within the cell itself, they have different states that they can undergo. So some of them are gates where they open and close depending on the concentrations of certain molecules outside and inside of the cell, and some of them have on and off structures which can help with the cell function. So given that these proteins have different structures and then they undergo different transformations based on the environment in which they are, protein folding itself becomes an important process for drug discovery, so that if you know the right shape and the right structure of the protein in its current environment, then you can target the protein accordingly, and then you know where the binding spots are and then what kind of residues are accessible, and then you can get that. So that's the protein folding background. And it just to talk about the challenges for this, I already talked about the different states that a protein takes, or different conformations that a protein takes. The computational side of it is a very time consuming and expensive process. I'll talk about that in the later slides that come in. But given that a larger protein, computationally simulating a larger protein would usually take a longer time, and then understanding different states also depends on everything around the environment. It becomes difficult to have a right model where you can simulate or you can estimate the right structure of the protein, which gets you one step closer to the drug discovery problem, because structure is everything. So once you know the structure, you can start looking at the right compounds to target the protein itself. So this is the overview of how AI has been involved in the protein folding problem. So the gold standard is still experimental methods. So you have experimental methods that are used to know the crystal structure of the protein, and then that becomes that is still a golden standard where people would take that as the best known structure. Which is obviously very time consuming and expensive. AI-based methods and physics-based simulations have been used for quite some time, where you're able to estimate a closer or a relatively inexpensive version of the structure, which you get from experimental methods. The first significant breakthrough came when DeepMind released their first AlphaFold model. That's when they were able to predict the performance or predict the structures to a level which was much higher than most methods that were entering the competition at that point of time. And then the second iteration of AlphaFold, which came around in 2018. So that's when the second AlphaFold model came in. It had much better performance overall. It included all the techniques that were having a lot of success at that point of time. So it used attention, and then it used a structure. I'll talk about the AlphaFold 2 model more in detail in the later slides, but they used all the components that were working well for large sequences, and that made AlphaFold 2 work well for most proteins. They showed a significantly high performance for single proteins, or like let's say monomers in general, and then the multimers had certain issues, but there's a lot of community effort where there were different modules that were built around AlphaFold 2 that was able to predict the multimers also in a better way. Since then, there have been a lot of other fold-based models that came in. You have Rosetta Fold, ESM folds, and the different versions of it. The most recent version, I think sometime in May this year, was the AlphaFold 3 version that came out. The major difference with AlphaFold 3 is that it is able to incorporate a lot of other molecules rather than just the amino acid residues itself. Because until AlphaFold 2, what you could do was you could just model the protein itself. So any other cofactors or any other small molecules or any other like DNA and RNA that would bind to the protein itself for it to have the final structure, you could not consider those other compounds into the mixture itself. So AlphaFold 3 has changed it by adding a lot more capability in that scenario. So this is the right time to mention this, because I think this is something that shows how important this problem itself is by people who were involved in protein design and then protein structure prediction as such have been they won the Nobel Prize in chemistry, and then that shows us how important this problem is, and then how this is going to change the research itself in the near future. So now I'll talk about AlphaFold. So I'll talk about the high level structure as such, because AlphaFold itself is a system that consists of multiple blocks, and all of them work in synergy to get you to the right structure. So at a simple level, so you have the input sequence, which is the amino acid sequence, and then you pass that in, and then you get the final 3D structure, which is your PDB file that you have for the protein. But to break it down into modules, so you have the input representation module, which you can see as the left-hand side of the red line here. So this is the input representation module. And then you have the major, the trunk of the model, which is called the evoformer. So that is where most of the modeling happens for the structure prediction. So once the model, the sequences and the representation passes through these models, it moves into something called a structure module, which is the tail end of the modeling, and then you relax the structures. And there is one additional component called recycling, where you take the prediction that comes out of the structure module, and then you recycle it as the representation for the next modeling section. And then that way you are able to build on top of a relatively you take something that is relatively like decently folded, and then you give that as a reference, and then you iteratively build on top of that. And that has shown to have a lot of better performance in building some complex proteins. So that's the high level block-wise structure of AlphaFold. So in Quantify, we have been using this, and then we have also like started doing a lot of customizations into this to be able to get the kind of performance that we want, and also bias the predictions towards something that we want, which is away from the default behavior of AlphaFold. So I'll talk about that right now. So this is where most of the modifications that we do take place. So it's in the input representation module. So there are three key places where we do that. So there is your positional encoding, which is required because these are sequences, and then you need to know the order of the sequences to send it to the attention module. So you need a positional encoding that happens as the first part. And then you have templates, and then multiple sequence alignment that is also sent in as input representation. MSA is required because the AlphaFold modeling takes evolutionary cues from MSA. So it knows, or it looks at row-wise and column-wise residues, and then it understands which residues go hand in hand. So you know that they are relevant for the function. They're also like closer because they probably either have similar mutation rates, or they, so based on that, it gets a first hypothesis of how the structure should look like. And then it also takes in templates. These templates are structures that are homologous, or you have structures that are relatively resolved in open source, which don't have decent resolution, but probably not the best resolution that you want to use for experimental experiments downstream. So you take those as templates, and then this gives you something called a pair representation, where you know how two amino acids are related to each other in like a matrix, and then you have the multiple sequence alignment, which gives you evolutionary information. So these are the three places where we use most of our customization. I'll talk about the customizations in the next slide. So based on what we understand from these modifications that we can do from this is how we have designed our AlphaFold workflow. So this is what we do. We start with any protein that comes in, whether it comes from a project that we do, or whether we run it as a research effort within the Quantify. So we start with a literature review, where we look at the open source structures that are available, homologous structures, understanding the binding parameters itself, and then what kind of the binding partners that the protein has, because like I said, depending on certain binding, the proteins also have different shapes. So we take all of that into consideration. We start with our we take our initial open source structures, and generally we start with the default AlphaFold prediction, whether it's a monomer or multimer. We start with the default prediction because that becomes our baseline. And once we get that, we check the RMSD and the structural parameters in terms of like the bonds that it should have. There are certain bond distances that is expected to be obeyed. So we look at those. That's the initial evaluation that we do. And for most monomer proteins, this step should be fine, because it AlphaFold does really well for those single proteins. But wherever you have a multimer, which is a little bit complex, that's where you need to like start adding in a little bit of customization. So usually once the evaluation is done, we if the evaluation passes the criteria, we just take that as the standard, and then we go to downstream experiments. But in most cases it doesn't, and that's where we start with the MSA and template analysis. The three main approaches that we usually take are: we bias the MSA. So usually MSA is, well, what we try to do is we cut down the total number of sequences that come up in MSA. That way we give a shallow MSA, so that the evidence in certain positions of these amino acid residues is not as strong, and then the model has an ability to try to come up with new hypotheses around those residues, because the deeper the MSA is, the more confident the model becomes in certain positions. And when you have open and closed structures, if the MSA is if you have strong evidence that is coming from MSA, you tend to move towards only one kind of structure, and then you don't have an ensemble that is being predicted. So that's what we do with MSA, where we experiment with the depth of the MSA. in templates, we check the open and closed ones, and then depending on what kind of structure we're trying to predict, let's say we're trying to predict the open structure, we make sure the templates consist of these open structures so that it gets the pair representation from open structures, and it can build a backbone based on that. And there's also recycling, which we take into consideration because, like I said, the whole purpose of recycling is you take a hypothesis that is built from our initial iteration, and then you pass it back to the model to build on top of that. And usually the default is set around like 3, but you can keep changing it depending on how complex your structure is, and then what kind of modification that you need to make to that. So these are the key iterative optimizations that we do. And once we do these iterative optimizations, we move towards relaxation and understanding the stability of the prediction itself, which is done through MD simulations, which I'll talk about once I go to the MD simulation set. So talking about MD simulation, so this is a physics-based simulation, and then it is used to understand how a system evolves from a certain time period X to a certain time period X plus whatever you take within the parameters of the simulation itself. It helps to understand how these dynamic molecules move, because like I said, proteins change in terms of like the kind of environment that they are in. They move from open states to closed states. There is also the small molecules that bind to that, which could trigger some conformational change, or they could just cause some changes within the protein that are of biological significance for you to come up with the right therapeutic. So these are the kinds of evaluation that we do with MD simulations. MD simulations is also used to predict like the structure itself, like from scratch, but they usually take like longer time scales, and anything that's after like a few thousand nanoseconds becomes hectic in terms of how much time it takes for the whole simulation itself. So we basically use AlphaFold to predict the structure, and then we validate their stability, and then we also validate protein ligand simulations, and then other downstream capabilities through MD. So this is the block level understanding of what happens within MD simulation. So briefly speaking, so you have to set up the initial environment. So that is the system setup that you have. So your proteins could just be with isolated, or it could be like a membrane protein where you have to pack the protein with the membrane, and you have to ensure the right orientation and everything. And then you also have to make sure the pH and everything is right, and the residues are in the right pH environment. So that's what you do in the initial setup. Once you do that, you do energy minimization to make sure like you are bringing the energy of the system down, and then you are making anything that could happen in terms of clashes could be taken care of. So that's when once all of the system is equilibrated, that's when you move to the production phase, where you run the system typically for a few hundred nanoseconds or 1,000 nanoseconds, depending on what kind of process you're trying to simulate. More often than not, we are trying to check the stability of the system itself. So we run it in an order of few hundred nanoseconds. But if you're trying to understand the motions of the protein based on how it moves from open to closed, depending on a certain ligand binding and all of that, you could move towards a longer time scale. So that moves to our pipeline. So this is how we have built our pipeline based on both what we have learned from AlphaFold and also molecular dynamics simulation. A couple of use cases to talk about, which would give this workflow a little more context, is we've used this more often than not in trying to understand how a protein could be engineered for better stability and better binding affinity, so that you know how what kind of modifications you can make, let's say in antibodies, or even within different mutants of the protein itself to understand the binding affinity. So we start with the input protein sequence, and we pass to AlphaFold. And like I showed in the previous slides, we have that whole iterative flow where we take the initial prediction, and then we iteratively make it better by making changes either within the MSA templates, or by adding positional encoding and the recycling factor itself. So once we do that, we get to our initial prediction for AlphaFold. And once we get that, this is where we are trying to understand the downstream effects, right? So now in terms of understanding the mutations, so what we know from AlphaFold is that from AlphaFold 2 mainly is that you can predict the structure, but whether when you make a certain mutation, whether that mutation is something that could, let's say, be a destabilizing one, which could basically make the whole protein unstable or not, is something that you would not know within the pipeline itself. You'll have to do an MD downstream to understand if the predicted structure is stable. So that's where we bring it into MD. So we simulate it for a couple hundred nanoseconds, and then we see if the overall RMSD of the protein is within limits, and then you know that this mutation is not something that is destabilizing, and it is a stable enough protein structure. So that's where we know that the mutations that we're making into these proteins are stable enough, and then we take that and then we start our protein ligand simulations downstream from that. So we can use different types of MD simulations there. But I'm just keeping it to classic MD simulations just to keep it brief. But that's typically how we get there. And once we have the trajectories of the protein ligand simulations that we have, and these ligands could be like small peptides or just like small molecules, we move to the binding affinity calculations from MMGBSC or PBSC calculations, which give us an understanding of how the mutation that was made in the protein is affecting the binding affinity for the ligand that it's supposed to interact with. And that gives us a better understanding of the overall landscape, and we can keep making changes accordingly, and then we can run the system at scale, which helps us in understanding the protein engineering. So I'll move to the last sections. Like I said, so this is where we mostly use it. So a couple of things to probably highlight are like in antibody design or antibody engineering, where you know the scaffold or the main structure, and then you want to make changes to that. So this is how you start making changes. You make single point mutations, and then you predict the structure, and then you can get the binding affinity of that. You can also make multiple mutations because AlphaFold does not differentiate between whatever mutation you make, right? So earlier, if you are making those changes within like PyMol or other softwares, you probably make single point mutations because most of the single point mutations do not really change structure. But if you assume that the structure could change, and then you want it to be predicted, then you can use AlphaFold to predict that, and then you can move downstream from there. You could also do protein design from by understanding like which areas are more conserved and then which areas are not conserved, and then you can like start mutating them combinatorially, and then you can still predict them and then get through the overall the pipeline. Yeah. So the key takeaways. So from the system that I talked about, which includes both MD simulations and AlphaFold, a couple of things that we have seen is that we are able to scale it effectively to different simulations. So we run it usually on cloud. So more often than not, we done this in GCP, but we could do this in other clouds as well. But we use cloud. We use these clusters to set up the MD simulations so that we run them in parallel. And then we also use open source softwares that have been optimized for GPUs and other hardware to make it seamless and then run without a lot of issues within the pipelines. Most of the modifications that we have made ensure that we are able to reach as close to the known crystal structures as possible, because like I said, AlphaFold in its default state has a certain kind of prediction, which might or might not be suitable for the kind of application that you are looking for. And all these modifications get you one step closer to the final step that you are trying to get there. I also talked about positional encoding. I think a key thing in positional encoding is most of these proteins are not cyclists, right? So they have the N-terminal and C-terminal, which is free. But if you want to model peptides, which are like smaller than 20 residues or like 30 residues, and then these are like cyclists, that's where you add the positional encoding to get this flexibility, because again, like the traditional or the default pipelines for AlphaFold, it tends to suffer when the protein length becomes smaller. And that's where you add the positional encoding, and then you bring the cyclization and the template so that you could also predict cyclic peptides, which are also an important class of therapeutic. So that's the next point where you see the lesser than 10 amino acid kind of challenges. The MD enhancements that that I talked about is something that gives you an additional layer of evidence for different kinds of protein engineering that you might do. And then once you do that, you need to have the experimental or at least this in silico evidence for you to understand that these mutants are making sense and they're actually stable for you to take them downstream for any other changes that you want to make. Yeah, I think that's the overall pipeline that we have built, and I think I'll be open to questions right now.
All right, thank you so much, Pali, for that insightful session. It's amazing how far our understanding of proteins and interactions have improved with the use of AI. We do have a few questions, couple of them from the audience. One is, since AlphaFold is MSA dependent, how do we modify your pipeline? Can I take that?
Yeah. So two answers. So now the first one is how to make modification within the AlphaFold 2 itself, right? So I talked about how you make modifications to the MSA pipeline, where the deeper the MSA is, the more because of how AlphaFold 2 is built with an MSA heavy backbone, the deeper the MSA is, the more evidence it takes from MSA, and then it becomes a little difficult to shake that MSA evidence out of it, which is why we cut down the evidence from MSA, and also we add the right templates to make sure you get a good balance out of that. The second part of the answer is AlphaFold. The DeepMind developers, have realized that there is an increased reliance in MSA for AlphaFold itself. So if you see the AlphaFold 3 paper that came out like recently, you'll see that they have drastically decreased the overall dependence of MSA within the overall pipeline. So they made it a pair representation heavy pipeline where you still have MSA evidence that comes in, but instead of having the Evo former block where MSA representation is being continuously updated to the pair representation throughout the 48 blocks, they basically have it very in the beginning itself, and then everything else is built on pair representation. So the newer ones are much better in terms of giving you a better structure, because MSA tends to heavily bias your structures.
All right.
So how
We're
scalable are the MD
almost out
pipelines?
of time, but yeah, like one last question. How scalable are these MD simulation pipelines? Can you answer that,
Simple answer
quickly?
is yeah. The simple answer is it depends on the cloud budget that you have, because these have been built to run seamlessly on whatever infrastructure you have. We have been running like 1,000+ simulations that we have in our pipeline, because those were the mutants that we wanted to simulate at a single point of time. And like I said, these are all. I mean, as much as we have built optimization in, MD is always a expensive process. So it just depends on how much resources that you have in your cloud environment. The more you have, the more you can scale it, because the pipeline is built to scale accordingly.
All right, thank you. And we have someone in the audience who love your headphones. Anyway, so that's all the questions we have for now. Thank you, Pali, for your time. Thank you, participants, for your questions. We'll now jump on to our next session, which is generative AI in pharmacovigilance. We're already up for that. So I hope to see you all soon there. Thank you.